Hi, and welcome back to my channel, Learning Biology with Dr. Vanessa, where I take difficult biological concepts and make them easy to understand. In our last video, we discussed the sarcomere, the functional, repeating unit inside of skeletal muscle. And we looked at all the important players like actin, myosin, and the bands and zones that give skeletal muscle its striated appearance. But knowing the parts is just the beginning. Today, we're going to put it all together and see how those structures actually work to make a muscle contract. This is where the famous sliding filament mechanism comes in. It's one of the most elegant examples of form meeting function in biology. Tiny proteins working in perfect coordination to create movement on a scale big enough to let you walk, lift, or even just blink. Muscle contraction happens because the thin filaments slide past the thick filaments. It's not that the filaments themselves shorten, they keep their length. Instead, the sarcomere shortens because the filaments overlap more. How does this sliding occur? It all comes down to the myosin heads on the thick filaments. Think of them like oars or hands. These myosin heads reach up, attach to the actin in the thin filaments, and then pivot, pulling the thin filaments inward toward the M-line, the center of the sarcomere. Each pull is small, but because billions of myosin heads are working together, the effect is powerful. The sarcomere shortens, the muscle fiber shortens, and ultimately the whole muscle contracts. That's the sliding filament mechanism in its simplest form. Myosin heads repeatedly attach, pull, release, and reset, walking along the actin filaments to generate contraction. Now, let's take a look at how it all happens. To begin, let's set the stage. Inside a resting muscle fiber, the binding sites on actin are blocked by a protein called tropomyosin. Tropomyosin is held in place by troponin, like a gatekeeper. When an action potential travels down the sarcolemma and into the T-tubules, it triggers the sarcoplasmic reticulum to release calcium ions into the sarcoplasm. Calcium binds to troponin, which shifts tropomyosin away and exposes the myosin binding sites on actin. Once these binding sites are exposed, the contraction cycle begins. Myosin heads are the workers of contraction. Each myosin head has an ATP binding site that functions as an ATPase. An ATPase is an enzyme that hydrolyzes ATP into ADP and a phosphate group. When ATP is hydrolyzed into ADP and inorganic phosphate, the myosin head is energized and ready to bind to the myosin binding site on actin. With actin's binding sites now exposed, the myosin head attaches to actin. This is called a cross-bridge. It's the first physical link between thick and thin filaments. Once the cross-bridge forms, the inorganic phosphate is released from the myosin, and the myosin head pivots, pulling the thin filament towards the M-line. This is the powerstroke. Imagine rowing a boat. Each pull of the oar moves the boat a little further. Similarly, each powerstroke shortens the sarcomere just a little bit. ADP is then released from the myosin head, completing the stroke. In order to reset for another powerstroke, a new ATP molecule must bind to the myosin head. This binding causes the cross-bridge to detach. The myosin head is released from actin. Without ATP, myosin would stay stuck to actin, which is exactly what happens in rigor mortis. When ATP is present, it's hydrolyzed again, re-energizing the myosin head and resetting it for another cycle. This sequence, attach, pull, release, reset, repeats as long as calcium and ATP are available. As millions of myosin heads cycle through this process in synchrony, the thin filaments slide deeper into the A band. This causes the sarcomere to shorten, the I band and H zone shrink, and the Z discs move closer together. Importantly, the A band doesn't change because the thick filaments themselves stay the same length. muscle contraction is really about the sliding of the thick and thin filaments over one another. So, the sliding filament mechanism is powered by ATP, regulated by calcium, and carried out by countless myosin heads working in concert. It's like your own microscopic rowing team, tirelessly pulling actin filaments to bring about movement. When a muscle fiber is relaxed, the concentration of calcium in the sarcoplasm is very low. Once the action potential moves along the sarcolemma and into the T-tubules, it causes the sarcoplasmic reticulum to release calcium into the sarcoplasm, which triggers a muscle contraction. The sequence of events that links excitation, a muscle action potential, to contraction, the sliding of the thin and thick filaments, is referred to as excitation-contraction coupling. Excitation coupling occurs when the action potential reaches the sarcoplasmic reticulum via the T-tubules. Here, there are two groups of membrane proteins, voltage-gated calcium channels and calcium release channels. The voltage-gated calcium channels serve as voltage sensors that in turn trigger the opening of the calcium release channels. The voltage change that occurs when the action potential reaches the sarcoplasmic reticulum is what triggers the voltage-gated calcium channels. Once these channels are opened due to excitation, or the muscle action potential that reaches the sarcoplasmic reticulum through the T-tubules, large amounts of calcium flow out from the sarcoplasmic reticulum into the sarcoplasm and surround the thick and thin filaments. This allows the calcium to bind to the troponin, which shifts tropomyosin away, exposing the myosin binding sites on actin. Now, myosin heads can attach and the cross-bridge cycle begins, and the muscle contracts. This will continue as long as muscle action potentials are generated. Once the last action potential has traveled through the T-tubules, the calcium channels will close. The strength of a muscle contraction depends on the starting length of the sarcomere before contraction begins. As the sarcomere is stretched, the overlap of the thin and thick filaments becomes less and less. With fewer points of contact, fewer myosin heads can bind to actin, which means fewer cross bridges form. As a result, less force is generated. So the more the sarcomere stretches, the less overlap there is, and the weaker the contraction becomes. On the other hand, as the sarcomere shortens, the filaments begin to overlap too much. Myosin quickly runs out of room to pull, and the thin filaments can actually interfere with each other's movements. This overcrowding also reduces the amount of force that can be produced. At an optimal sarcomere length, usually very close to the resting length found in the body, there's just the right amount of overlap between actin and myosin. This allows the maximum number of cross bridges to form, generating the strongest contraction. So we've seen now how the sliding filament mechanism explains muscle contraction, from cross-bridge cycling to excitation-contraction coupling, and even how the length-tension relationship impacts force. But the question remains, how does an action potential actually propagate through the muscle cell in the first place? This is all explained by the neuromuscular junction, which we'll dive into in the next video. If you enjoyed the content of this video, please make sure to subscribe to this channel, give this video a like, and turn on notifications so that you never miss out on new content. Thanks for watching, and I'll see you in the next video.